Class 11th. Today we will be discussing about dark reaction in this video. The dark reaction, as you can see, it is also known as biosynthetic phase, right? Because here the synthesis of a natural food product, sugar, is produced in the form of glucose by the plants. And you know that till now we have completed the events of light reaction with respect to activation of chlorophyll, photolysis of water, and the end products were ATP by phosphorylation and NADPH by reduction. And we know very well that when we enter this dark reaction, those two products, the ATPs and NADPs, are supposed to be fixed because NADP is reduced, so NADPH, this is what you get as end product of light reaction. That means both of these are going to be used in this dark reaction. So you may write it this way, that the end products of light reaction are going to be used in dark reaction, right? Just keep this in mind. Here, before we start straight away with the the cycle of dark reaction, you have to keep one thing in your mind, that the dark reaction may be known as, this is also known as Blackman's cycle or Blackman's reaction, the way you wrote Hill's reaction for light phase, right? Light dependent phase. This is Blackman's reaction for light independent phase. That these are the different names by which a dark reaction may be called. So here you can see that I have written two cycles here, Calvin cycle and Hatch-and-Slack cycle. What you have to see is, it doesn't matter which cycle you are talking about, ultimately Calvin cycle is going to happen. In Calvin cycle it is automatically happening, right? In Hatch-and-Slack cycle also, doesn't matter what different procedure is followed, but ultimately it is going to come to Calvin cycle only. So C3 cycle is supposed to be followed later on, but maybe a different path is followed, that's why you are calling it C4 cycle. Okay, so always remember this, that C3 cycle occurs, or the steps of C3 cycle are going to be there in both types. It's just that I'm going to tell you the difference now. What is the difference between these two cycles? The difference is actually maintained because of the first immediate product which is formed, right? So suppose during dark reaction, always remember the dark reaction starts with the base compound, that is RUBP. Hold on. That is RUBP. RUBP is ribulose bisphosphate. Ribulose bisphosphate, right? So here this RUBP is a 5 carbon atom compound. Okay, so during dark reaction the atmospheric CO2 is supposed to be fixed, right? So you see that here 5 carbon atom, RUBP is 5 carbon atom compound and CO2 has 1 carbon. So here the ultimate compound that is formed should be a 6 carbon atom compound, isn't it? 5 from RUBP and say 6th one from CO2. This fixation of CO2 is basically known as carboxylation. It is an enzyme mediated process, but ultimately the product which is formed is PGA. This word is heard by you in class 10 too. So this PGA is phosphoglyceric acid. Phosphoglyceric acid. This is the first immediate product which is formed in C3 cycle, and why this product is basically responsible for naming the whole cycle as C3 cycle, the dark reaction, because this PGA is a 3 carbon atom compound. Right, so first immediate product formed after carboxylation, after fixing the molecule of carbon dioxide, it is a 3 carbon atom compound, and that's why the whole cycle is known as C3 cycle. This is one type. You might be wondering that here see 5 carbon atoms from RuBp, 6 from CO2, so it should have been 6 carbon atom compound straight away, but this is not so simple. PGA is not a 6 carbon atom compound, it is 3 carbon atom compound. So how to divide? We always write it as 2 molecules of PGA, right? So 3 plus 3. Okay, this is how 6 carbon atoms are fixed. Once you know this, now you come to the second cycle. What is the difference there? There also you have you don't have RuBp there as the primary base molecule. There you have phosphoenol pyruvate, right, PEP. And here when carbon dioxide is fixed, the first immediate product which is formed is, so ribulose is replaced by PEP and carbon dioxide is fixed similarly, but the first product form is OAA, that is oxaloacetic acid. Oxaloacetic acid, right, and it is a 4 carbon atom compound. So that means if a plant is following a pathway for dark reaction where 4 carbon atom compound is formed at first, that will be the C4 cycle. You also call it Hatch and Slack cycle. And another one is, the main one is rather most abundant one found in all plants, that is say C3 cycle, where the primary molecule which is processed is a 5 carbon atom compound RuBp, right. In this video only I am letting you know about the bioenergetics also. That means during 1 Kelvin cycle, how many, I mean you have to talk about the ATP usage and NADPH which are used in this. So always remember 3 ATP and 2 NADP in one cycle, in 1 Kelvin cycle it is used. And we know very well that 1 Kelvin cycle is responsible for fixing one molecule of CO2, and in totality to make C6H12O6 you need 6 molecules of CO2 to be fixed, right, 6 molecules of CO2 to be fixed. So that means for making a sugar molecule, a glucose molecule, this Kelvin cycle should be repeated 6 times, right, and in one time 3 ATP and 2 NADP. So for making one glucose molecule you have to multiply them by 6, so 18 ATPs and 12 NADPs, these are used in making one molecule of glucose, right. So this is just a brief introduction about that what are C3 and C4 cycles, and then what is the bioenergetics, I mean how many ATPs and NADPs which are coming from light reaction, they are used in where they are fixed basically in this dark reaction. Okay children, so if you have any issues with this particular portion, you please feel free to ask me. Thank you.